Welcome to G Fun Facts Online. Today, we are diving deep into a moment that redefined science in the early months of 2026. For a century, physics and biology were treated as separate kingdoms. Physics dealt with the cold, hard laws of the quantum universe, while biology dealt with the warm, messy complexity of life. But recently, that wall came crashing down. We are going to talk about the protein qubit. This is not science fiction. This is the story of how scientists managed to encode a functioning quantum bit, not in a silicon chip, but within the organic architecture of a fluorescent protein. We will explore how this happened, the physics of entangled amino acids, and what this means for the future of living quantum computers and our understanding of human consciousness. Let's get right into the science. To understand why this is such a massive deal, we first need to look at the silicon wall. For the last few decades, if you wanted to build a quantum computer, you had to build a freezer first. We are talking about dilution refrigerators that hum in stark white rooms, cooling superconducting chips down to temperatures colder than deep space. The reason is isolation. Quantum states, like superposition and entanglement, are incredibly fragile. Thermal noise, electromagnetic interference, and just the vibration of atoms can cause a qubit to decohere. Decoherence is the enemy. It is when the magic of quantum mechanics collapses into mundane classical physics. So the strategy has always been to isolate the machine from the environment. Biology is the opposite. It is warm, it is wet, and it is chaotic. A living cell is a storm of molecular motion. To a classical physicist, a cell is the absolute worst place to try and perform a quantum calculation. But nature had a secret. While we were fighting to isolate qubits, evolution was designing systems that thrive specifically because they interact with their environment. The breakthrough came in August 2025, when a team at the University of Chicago Pritzker School of Molecular Engineering shattered the thermal barrier. They didn't build a qubit, they grew one. They used a genetically modified version of a fluorescent protein, very similar to the green fluorescent protein, or GFP, that we originally got from jellyfish. They created a molecular structure that could host a coherent spin state at room temperature. This is the protein qubit. It is a dynamic, foldable, genetically encodable machine. It represents the first operational quantum bit that can be synthesized by the very cells it is meant to measure. So how does a piece of protein act like a quantum computer? We need to look at the anatomy. The core of this technology is the fluorophore. This is the chemical structure within the protein that absorbs and emits light. In a standard GFP molecule, this is a barrel-shaped cage made of beta sheets. Inside this cage is a chromophore, formed by three specific amino acids: serine, tyrosine, and glycine. The researchers modified this structure to stabilize what is called a triplet state. In standard quantum mechanics, electrons usually pair up with opposite spins, one up and one down, canceling each other out. This is a singlet state. But, using specific wavelengths of laser light, researchers can kick an electron into a higher energy orbit, and crucially, flip its spin. This creates a pair of electrons with parallel spins, which is the triplet state. This state acts as the qubit. It has three sublevels that can be manipulated using microwaves. Because that beta barrel structure acts like a Faraday cage, shielding the chromophore from the water molecules outside, the spin state can maintain coherence for microseconds. In the quantum realm, a microsecond is an eternity. It is long enough to perform logic operations or precise sensing measurements. But the real innovation wasn't just the single fluorophore, it was the environment provided by the surrounding amino acids. This leads us to the concept of entangled amino acids and tryptophan meganetworks. You see, proteins are not just structural scaffolds. They are conductive wires for quantum information. The amino acid tryptophan is the star here. It has a large double-ringed indole structure that is aromatic. In chemistry, aromatic means it has a delocalized cloud of electrons, floating above and below the ring. When you arrange multiple tryptophan residues in a specific geometry, like a spiral staircase, their electron clouds interact via dipolar coupling. If the spacing is just right, they stop behaving as individual molecules and start behaving as a single quantum entity. This leads to a phenomenon called superradiance. Think of it like a stadium full of people. If everyone claps randomly, you hear noise. That is decoherence. But if everyone claps in perfect unison, the sound is amplified exponentially. That is superradiance. When a network of tryptophan molecules is excited, they form a collective state. They absorb and emit photons not as individuals, but as a giant megamolecule. This collective behavior protects the quantum state. If one amino acid gets kicked by thermal vibration, the energy is instantly redistributed across the others, preventing the information from being lost. This is how biology survives the warm, wet environment. It finds safety in numbers and geometry. There is another layer to this architecture, and it involves chirality. Biology is homochiral. Life uses almost exclusively left-handed amino acids. For years, we thought this was just a quirk of evolution. Now we know it is a functional requirement for quantum efficiency. This is due to the chiral-induced spin selectivity or CISS effect. It dictates that when an electron moves through a helical molecule, its spin becomes coupled to its momentum. Basically, a right-handed helix might only allow spin-up electrons to pass. It acts as a highly efficient spin filter. In the protein qubit, the helical structure acts as a built-in initializer. It ensures that the electrons entering the fluorophore are already polarized. This removes the need for the massive external magnets usually required to align qubits in classical quantum computers. Now, let's talk about what we can actually do with this. The most immediate application is biosensing. Traditional quantum sensors, like nitrogen vacancy centers in diamonds, are powerful but invasive. Inserting a diamond nanorod into a cell is like tossing a brick into a washing machine. It disrupts the system. The protein qubit, however, is a Trojan horse. Scientists can splice the DNA sequence for the protein qubit into the genome of a cell, like a neuron. The cell's own ribosomes then manufacture the sensor and place it exactly where it is needed. Once inside, the protein qubit acts as a spy. Its triplet spin state is extremely sensitive to magnetic and electric fields. For example, we can perform nanoscale (MRI). By measuring the shift in the spin frequency, researchers can detect the magnetic fields generated by the firing of a single neuron. This allows for network-level mapping of brain activity with single-cell resolution. We can also map electric fields. The voltage across a cell membrane is the battery of life. Protein qubits can measure these fields with sub-millisecond precision. We can even measure temperature. Quantum states are very sensitive to heat. The protein qubit has revealed that mitochondria, the power plants of the cell, run significantly hotter than the rest of the cell. This implies that biological thermodynamics is far more complex than we thought. Another application is entangled two-photon microscopy. One of the biggest problems in microscopy is that to get a bright image, you have to blast the cell with high-intensity lasers, which eventually kills it. But if you use entangled photons, partners that are quantum mechanically linked, they arrive at the molecule together. The probability of them both being absorbed is exponentially higher. This means you can get the same bright image using a fraction of the light intensity. The protein qubit is designed to be a superabsorber of these entangled photons, allowing us to film biological processes for days without damaging the sample. This technology also brings us to the frontier of quantum biology and consciousness. You might have heard of the Orco-R theory proposed by Roger Penrose and Stuart Hameroff. They hypothesized that consciousness arises from quantum computations inside microtubules within brain neurons. For decades, this was dismissed because microtubules were considered too noisy. But the discovery of superradiant tryptophan networks has reignited this debate. We now have proof that protein structures can maintain quantum coherence at physiological temperatures. The tryptophan networks in microtubules are strikingly similar to the engineered arrays in protein qubits. Further evidence comes from anesthesia. How does a gas like xenon switch off consciousness? Recent experiments show that many anesthetics bind specifically to the hydrophobic pockets in proteins where tryptophan networks reside. When they bind, they disrupt the electronic environment, effectively detuning the quantum network. If the brain relies on superradiant quantum channels for consciousness, and anesthetics cause decoherence in those channels, then the loss of consciousness is literally a quantum crash. The protein qubit gives us the tool to test this directly. Finally, let's look at the future. We are moving toward the living quantum computer. By the late 2020s, we might not just be reading biology, but writing with it. Imagine a bacterial colony where each bacterium contains a protein qubit. These bacteria could be connected via photonic nanowires or entangled photons. This would be a massive, self-replicating quantum processor. It wouldn't be fast like a silicon chip, but it would be massively parallel. And this leads to the bioquantum internet. Protein qubits are optically addressable. Imagine a medical implant that monitors your blood chemistry with quantum precision, entangles its state with a photon, and teleports that information to a medical computer. It could detect a single cancer cell hours after it appears. The protein qubit is more than just a sensor. It is a bridge. For the first time, we have a technology that speaks the native language of both quantum physics and biology. By harnessing the entangled amino acids that nature has spent billions of years refining, we are unlocking a new era. We are learning that life is not just a chemical process, but a quantum one. The protein is no longer just the building block of life. It is the building block of the future's information technology. That is all for this deep dive into the protein qubit. Thank you for listening to G Fun Facts Online.